Hey,
Hi, how are
hi,
you?
fine.
Just
So
want
it's
a quick tech check from my side. So clearly I'm audible, so that's good. Can I
Akshatha,
just
hello,
try
sir. Yes, yes, we please share.
the screen and just check? Oh, I think it says disable
Yeah,
screen share.
just 1 second. Now you can share, please.
Okay, that works, right? And I'm just gonna make it a slideshow mode. Does that look okay?
Yes, yes. You want to
Okay.
try changing the slides once?
Oh, let me do that as well. Yeah, good point. Okay. All right.
Yes.
Uh,
Yeah.
uh,
So,
yeah. Thank you.
sir, would you like to share that we tried
mine
sharing the
also?
screen? Yes, sir.
Okay, why
Just
we can do that? I think you opened it for me, right? Yes. Does it come along?
ah, yes, sir. Perfect.
Let me see. Yeah, animations do work. Yeah, that's good.
Yes, sure, sir.
Awesome.
Yes, sir.
Thank you.
Thank you. Thank you, sir. Thank you. Thank you both for joining. Yes.
Sure.
We will just start like I think 4 minutes past 6. So we'll wait for people to join in. Then I will introduce. I will start. I will just give a brief introduction and then introduce you, and then you can take over. So first, the Dr. Neeraja will speak. Then afterwards, once her talk ends, you can. I'll introduce you, sir. Then again, you can start sharing your screen.
Brilliant. Thank you.
Hello everyone, thanks for joining. We'll wait for another 2-3 minutes for others to join and start shortly. So, good evening, everyone. Thank you for joining today's webinar. So, where we'll be exploring the key highlights and the groundbreaking updates from the IDweek 2024. So, for those who are not aware, IDweek 24, IDweek is a premier infectious disease meeting. So, it's a annual Infectious Disease Society of America conference. So, where it brings together the greatest or the largest research from all parts, including infectious diseases, microbiology, and public health. So this year it was in the Boston, and this year I had the opportunity to visit or be on site. So through that, one of the opportunity I had was good networking. So I could meet various people from India and also Indians from abroad. So one of them is Dr. Neerja Swaminathan. Thank you for being with us for now. Uh, so in this session, our panel will distill and try to cover the important or impactful takeaways from the meeting and share insights which can be relevant to your clinical practice. So let's dive into these updates. So again, I would like to reiterate that it was ID week is like around 4 days of extensive conference, and there are various halls and around 150 200 sessions, so everything can't be recapitulated in the short span of time. So we'll be recapitulating some important studies or which we felt it was interesting. So without any further ado, let's start with today's session. We have the first speaker, Dr. Neerja Swaminathan. She is a American board certified internal medicine and infectious diseases expert, and she is currently working as an assistant professor in University of Utah. Oh, she has received various awards to her credit, including IDweek Trainee Abstract Travel Award and Leo M. David of Society membership for outstanding achievement in teaching of medical students, as well as Best Clinical Research Abstract. So I would like to take this opportunity to first of all thank Dr. Neerja for making time. And there is a lot of time difference as well, I agree. So like my heartful thank you to you for accepting to be a part of this. And I would like to like over to you, Dr. Neerja, for taking the session. Thank you.
Thank you. Thank you so much for the introduction as well as the invitation to be here today. I've been following your channel for some time, so I know it's very high yield and really educational content. So I'm glad I can be part of it this time. So let me share my screen. All right. So I think, as Akshata mentioned, IDweek is a great place not just for the educational sessions, but all the opportunities to network and meet your colleagues. And I was glad I could see her in person after all the YouTube seminars and stuff that I have seen online. So I'm glad I could connect in person. And it's also great that you're doing this initiative so we can meet as a smaller group and continue to interact and hopefully collaborate in the future as well within our community. So for IDweek 2024 recap today, I'm sharing mainly transplant ID takeaways. As Akshata mentioned, I work at University of Utah in Salt Lake City in the US. And let's see. All right, for my disclosures, the only conflict is with my sleep schedule. It's 5:30 in the morning here, so definitely an earlier start to my weekend than usual. I take no credit for any originality. All the content here belongs to the IDweek presenters, so I'm just curating what I thought was the most high yield. Having said that, this was not an easy task because there were over 250 sessions at IDweek this year. And if you try and filter by transplant ID type, then there are at least 69 sessions. And I only have 30 to 40 minutes to recap. So I'm going to try and pick the stuff that is most, I think, resonated with me the most because it's something we see in clinical practice frequently. And I think that you will find that to be true for your practice as well. And I've included some screenshots from these sessions, but this is by no means comprehensive. It's a very broad strokes overview. But the good news is the full sessions are available on demand for a whole year, so please check them out online. So with that, I'm going to dive into the first theme that I think has been the theme of many IDweeks, especially in transplant ID, certainly, and that is antimicrobial stewardship. This population of transplant patients is definitely a challenging group in which to do stewardship. These patients are varying degrees of have varying degrees of immunocompromise. They, compared to the general population, have a higher risk of multi-drug resistant organism infections as well as colonization. And there's a higher exposure in this population to antibiotics and healthcare settings in general. And the data or guidelines that we have often will exclude these patients. In general, these patients are perceived as sicker or exceptions to the guidelines, therefore. And so primary providers often also, whether that's the surgeons or the hematology folks, might be unenthusiastic about de-escalating antibiotics in this subgroup. But because of these very challenges, these are the very things that make this population also ripe for antibiotic stewardship interventions. So one of the ways that was highlighted in some of the sessions to do this is surveillance for multi-drug resistant organisms pre-transplant. And the idea behind this is that instead of giving broad spectrum antibiotics to just every patient, especially, for example, in our solid organ transplant cohort, instead of giving daptomycin, meropenem, or some big gun antibiotics in just every patient that's pre-transplant, we try and identify the subgroup of patients that are at higher risk and will benefit from broader antimicrobial use. This way, we can be more judicious with our use of antimicrobials. And one of the ways people have done this is to look for intestinal colonization by doing rectal swabs. And in this next slide, I'm summarizing some of the studies in solid organ transplant patients that looked at this. So the first one here is Radcliffe et al. In 2022, who looked at liver transplant patients, and they were essentially screening for azole non-susceptible candida in order to tailor antifungal prophylaxis. So in the azole non-susceptible group, they would give tailored prophylaxis with an echinocandin, either anidulafungin or caspofungin. And in this cohort of the 48 transplant patients they had 10 who were colonized. 7 of these 10 got appropriate prophylaxis, and only 1 of the 7 who got prophylaxis developed candidemia. In the next study, they did something similar, but this time with CRE or ESBL screening. And this was heart and lung transplant patients. There were a total of 126, of which 12 were colonized. 10 of these 12 who were colonized with CRE or ESBL got targeted antimicrobial prophylaxis. And only one of those 10 went on to develop a bloodstream infection in the early post-transplant period. But both of the patients who did not get appropriate prophylaxis, and both happened to be colonized with Klebsiella-producing carbapenemase or KPC, both those patients ended up with bacteremias in the early post-transplant period. And then this last study looks at vancomycin-resistant enterococcus. So in a liver transplant population, there were 27 patients that were screened positive to be colonized with VRE, and the intervention was to give targeted antimicrobial prophylaxis with daptomycin. And 25 of these 27 got appropriate prophylaxis, and none of them went on to develop VRE complications. But both the patients who did not get VRE prophylaxis developed bacteremia in the early post-transplant period. And I think this is the first 90 days. So the takeaway essentially is that even though these are limited data, really small numbers, as you can see, 20 and 40 patients, it makes a case for trying to identify the high-risk population that benefits from more tailored prophylaxis. Certainly, this data is not enough to make any universal screening recommendations. And when to screen, how long to screen, what all microorganisms to screen for, these are all questions that still remain. But this is a way to, I think, at least identify some high-risk patients. A similar theme was explored also in our heme BMT population with febrile neutropenia. So this is a busy table, and we're not going to go through all of them, but essentially they were asking the question of, does intestinal colonization with resistant bacteria predictably predict subsequent bloodstream infections? And if you can see this table here, I'll draw your attention to this last conclusion column. It's very heterogeneous results. Some of these studies show association and others don't. Some of the ones that have a positive association of colonization with subsequent bacteremia development are in the patients that were screened for ESBL or CRE. So I think our takeaways from this can be, if you have known colonization with CRE or ESBL, then that might help to predict a subsequent bloodstream infection. But the information for something like Pseudomonas is scarce. Again, like we mentioned with the solid organ cohort, who to screen, which microorganisms to screen for, and how long to do this, all of this are certainly unanswered questions or inadequately answered questions that require further studies. So while we can't make universal recommendations, I think there is a case to be made for surveillance in certain high-risk patients. And if we have data regarding previous colonization or infection with multi-drug resistant organisms, we can use that to guide our antibiotic therapy. Another strategy that has been employed for antimicrobial prophylaxis in terms of de-escalation is the MRSA-NAIRS or methicillin-resistant Staph aureus NAIRS screening. I think this has been used a lot in the general and non-immunocompromised cohorts to de-escalate vancomycin use when negative, because it has a really high negative predictive value. But its applicability in the solid organ transplant or immunocompromised cohort had not been very well studied. We were extrapolating the guidance, but now there's a lot more data in this cohort as well. So this table summarizes some of the studies, and I think the largest one is this first one by Shaw and colleagues, and they looked at solid organ transplant, BMT, and neutropenic patients. And essentially, they were comparing MRSA nasal swab to positive bacterial cultures within 28 days of screening, and looked at the sensitivity, specificity, and negative predictive value. As you can see, it's a really high cohort of patients, over 600,000, and they found excellent negative predictive values even in this population. So what that means is, with a negative result, we can reliably de-escalate vancomycin use in these patients. And these two other studies also looked at a similar thing in AML patients and found 98, 99% negative predictive values. The other big topic within stewardship is about shorter courses of antibiotics. So is shorter better? I think this is a topic that, at least in the non-immunocompromised patients, has gained a lot of momentum. And I think more and more there is a case to be made for shorter courses in various clinical syndromes. but whether this can be extrapolated to our immunocompromised population is not very well studied. But there's a lot of upcoming literature on this. The things we know are that antibiotics cause incremental daily harms. Also, it has an impact on the microbiome, and that in turn can cause graft failure. But identifying which patients benefit from longer courses and who can get away with shorter courses is often tricky. So in some of the sessions that they highlighted specifically Pseudomonas and Enterobacterales bacteremia. So we'll talk about both of those. The first is Pseudomonal infections. So this table summarizes some of the studies that look at short versus long course antibiotics for Pseudomonal bacteremias. The first one by Faber and colleagues in 2019, it's a mixed population, but 65% of them were immunocompromised. And these were Pseudomonal bacteremias or bloodstream infections. As you can see, they've excluded a lot of deep-seated infections, complicated infections. And they were comparing short versus long course of antibiotics. Short was defined as 7 to 11 days, long 10 to 21. And the analysis was done through a weighted cohort with inverse weighted propensity score. So that's effectively a statistical tool to try and balance out the baseline characteristics in the two groups. And this is about 250 patients, 72 in the short arm, 170 in the long course arm. And the outcomes they were looking at is recurrence of Pseudomonas infection or 30-day mortality. And as you can see, it's very comparable, 14% versus 13%. This next study from Korea with Bay and colleagues, again, also looked at uncomplicated Pseudomonal bacteremia. 43% of them were immunocompromised hosts. The definition of short versus long, as you'll see in the studies I go through, are a little bit variable, but kind of similar in most of them, minor tweaks. Again, a weighted cohort analysis of around 300 patients, 97 in the short arm group, 192 in the long arm group. And they also looked at a composite of recurrence of bacteremia or 30-day mortality after discontinuing antibiotics and found very comparable numbers. This study from Babich and colleagues is a much higher number of patients, over 600 patients across 9 countries and 25 centers, and 22% of the population were notably neutropenic patients. And again, the same theme, the 30-day mortality and recurrence being similar in the short versus long arms. Interestingly, they, and not unexpectedly, I would think, they found that the length of stay was lower in the short course treatment arm as compared to the long treatment arm. And the two more recent studies from 2024 are Feng and colleagues in China, who looked at this specifically in hematology oncology patients and acute leukemia patients. Again, this is looking at mostly patients that have uncomplicated bacteremias, and they had a weighted cohort of 200 patients in each group. And the composite outcome of recurrence of infection in 30 days or death was comparable in both groups, and the same even at 90 days. And this last study by Truong and colleagues, this was not bacteremia, but actually Pseudomonal pneumonias. And they looked at drug-resistant pneumonia, where they defined it as resistance to even one agent like a fluoroquinolone. Interestingly, 15% of this cohort is solid organ transplant patients. They've excluded polymicrobial pneumonias or empyemas. And they compared short course versus long course, where the cutoff was 8 days. There were 30 patients in the short arm and 55 in the long arm. And when they looked at outcomes of clinical success, which is defined as resolution of symptoms and not needing any additional antibiotics for the same indication, they found similar results, kind of comparable clinical success between the short and long arms, even when adjusted for comorbidities. And the mortality at 30 days is also comparable. So the takeaways from all of this essentially is that for Pseudomonal bacteremias and infections, a short course might be adequate for a select group of patients, especially in those that have uncomplicated infections and source control is possible. If they are seen to have a good clinical response and you know that you can monitor them outpatient as well for recurrence of infections, all of these studies gives us some data to back up our reasoning to do shorter courses, even in our immunocompromised patients. But certainly, we do need larger cohorts especially for drug-resistant Pseudomonas. A lot of these studies did not have adequate representation of some of the more drug-resistant Pseudomonal isolates. The solid organ transplant group is definitely underrepresented. And as you can see in most of these studies, I was highlighting recurrence of infection and mortality as the outcomes, but the other aspect of antibiotic-related harms like IV-related complications or length of stay and some of the other patient-centered outcomes also need to be studied, because those are the things that will definitely impact the patient, and shorter courses might be better from that standpoint as well. Ultimately, it'll all come down to a nuanced approach, but having some data to back up shorter courses for a select group is definitely helpful. A similar theme was also seen with Enterobacterales bacteremia. So I've summarized 3 studies here in the table. The first one by Yahav and colleagues was a randomized control trial non-inferiority of over 600 patients. And this included some immunocompromised patients, as you can see. And these were uncomplicated gram-negative bacteremias. And they had 2 arms, 7 days versus 14 days. And they looked at a composite of 90 days of all-cause mortality, relapse, readmission, and other complications. And as you can see, very similar numbers, 45 versus 48%, not very statistically significant. So making a case for shorter courses, showing that 7-day is non-inferior to 14-day. Herrera and colleagues looked at the same thing specifically in neutropenic patients. So this was a prospective study of short versus long, where the cutoff was 7 days, 100 patients in each arm. And as you can see, again, recurrence and 30-day mortality, very similar in both. And the median length of stay notably was lower in the short treatment course. And Ranganath and colleagues looked at this in the Mayo Clinic, also in neutropenic patients specifically. And they had another arm of intermediate group as well, which was defined as 11 to 14-day courses. And there were about 200 patients total. And they also looked at a composite of all-cause mortality and relapse within 90 days. And as you can see, the hazard ratio adjusted shows that short is not inferior to intermediate or prolonged courses of antibiotics in Enterobacterales bacteremia. So again, in patients with good clinical response, no source control issues, generally short courses seem reasonably safe, even in our immunocompromised population. So that is some of the highlights of, I think, stewardship-related topics that I wanted to highlight. I think that's definitely very useful for our clinical practice. The next big theme that I want to talk about is CMV. I think no transplant clinical practice is without our fair share of CMV-related issues. And the talk I want to highlight is about CMV prophylaxis and what's new in terms of prevention strategies. So just a little background, CMV prophylaxis, for our solid organ transplant patients, the highest risk group for CMV is the D plus R minus group. And for the stem cell transplant, it is the R plus group. The other factors that increase risk of CMV are T-cell depletion, getting GVHD or rejection treatment, and all of those increase the risk of CMV as well. And the goal of CMV prophylaxis is essentially to prevent tissue-invasive CMV disease and direct effects of CMV. Also, it has some indirect effects like allograft dysfunction and increasing the risk of other opportunistic infections as well. So there are 2 broad strategies for CMV prophylaxis. One is universal prophylaxis, and the other is preemptive therapy or PET, wherein you essentially keep doing weekly surveillance, and once there is a pre-specified threshold that's crossed, you start treating them until viremia clears. And there's also a hybrid strategy that some centers employ where you do prophylaxis for a period of time and then you continue to follow with surveillance. In 2017, there was this study which authorized use of letermovir in patients with stem cell transplant. And it showed that letermovir significantly reduces clinically significant CMV infection and also had a mortality benefit in certain high-risk patients in the first 100 days post-transplant. So these are the different, this is kind of a background of, CMV prophylaxis, but we all know that despite giving prophylaxis, there are certain complications. One is that a lot of these patients develop late onset CMV disease when the prophylaxis is discontinued. And there can be antiviral resistance, resistant refractory disease that we see, and toxicity associated with the antivirals, not to mention some cost effects as well. So there are certain subgroups where they looked at CMV prevention strategies, and we'll talk about 3 of these. The first one is comparing PET versus prophylaxis in the high-risk liver transplant population. So this is a randomized controlled trial of over 200 patients where they essentially were randomized to 2 arms. One is prophylaxis for 3 months, and the other is preemptive therapy. And as you can see, there was a lower rate of 1-year CMV disease in the preemptive therapy arm, 9% versus 19%. The other aspect of this study is that they looked at the immune response at day 100 by CMV IGRA, or interferon gamma release assay testing. And as you can see, both for CD4 and CD8, the immune response is lower in the group that receives antiviral prophylaxis, and better CMV specific immunity is seen in those that receive preemptive therapy. So the takeaways are that at least in high-risk liver transplant, PET should be preferred over prophylaxis, but perhaps we should even reconsider our approach of doing PET for other organ groups, but certainly we need more data regarding that. And PET helps to achieve higher CMV-specific immunity. The next subgroup is the high-risk kidney transplant, and the goal is to reduce antiviral toxicity. So this RCT looked at letermovir versus valganciclovir as antiviral prophylaxis for the first 200 days post-transplant. And as you can see, they found letermovir to be non-inferior to valganciclovir when they looked at CMV disease up to 1 year post-transplant. It's quite comparable. And again, anyone that's in the field knows that the main utility of letermovir is the lower neutropenia and side effects seen. And that's exactly what they also showed in the study, that it is less toxic as compared to Valsite and has non-inferiority. The next subgroup is looking at extending duration of prophylaxis in the high-risk stem cell transplant population. So we already knew that letermovir is used for the first 100 days post-transplant for CMV prophylaxis, but there's a spike of CMV disease that's seen right when prophylaxis is discontinued. So this RCT looked at 220 high-risk patients of the stem cell cohort and looked at extending the prophylaxis to 200 days versus placebo. So as we can see, clinically significant CMV at 200 days was much lower in the letermovir cohort as compared to those that received placebo. But this difference kind of evens out as you go to time points 38 and 48 weeks. So as you go along, the difference is not so significant. They submitted some of their immune response studying in the ECMID conference, which happens in Europe annually. And they found that the patients that were receiving antivirals with letermovir had lower CMV-specific immunity again, as compared to those that were not. So what this tells us is that extending prophylaxis can reduce CMV disease while it's ongoing, but the risks of late onset disease will still exist. But giving prophylaxis does delay development of CMV-specific immunity. So what can we take away from all of this? Firstly, preemptive therapy is a strategy or a tool that we have to actually allow for permissive replication of the virus and develop CMV-specific immunity. And so we should think of prophylaxis, therefore, as a way to suppress CMV replication until the immune system can recover. How to achieve these two seemingly at odd goals of having permissive replication of CMV while also facilitating a durable immune control. I think there aren't too many current tools that are answering this, and we need newer tools for CMV prevention. And some of those were highlighted in the conference. One is that there are vaccines that are in the late stages of clinical development, which look very promising. There are several others that are also being looked at, but there's not a whole lot of data about it just yet. I think especially with monoclonals and CMV-specific viral T cells, there aren't any products on the market now that are in late stages of clinical development. So I'm just going to include a small word on the vaccines as well. There's two currently that are being looked at. The first one is a triplex vaccine, which is a modified vaccinia vector, and the other one is a Moderna vaccine, mRNA. And both of these, the first one has been completed in phase 2 trials, found to be in stem cell transplant patients, found to be safe and effective in reducing CMV disease. And they're trying to look at this in solid organ transplant as well. And Moderna mRNA just finished a phase 1 in healthy adults, and they're going to enroll for phase 2 as well. So that's exciting in the field. And that is all I have for today. These are some of the talks that I've highlighted today. I'd like to thank those speakers for really very great sessions. And I welcome any discussion or questions that the audience may have.
Thank you, Dr. Neerja, for recapitulating and trying to minimize and in such a small, short duration of time, you have highlighted the very important studies. And yes, these are clinically every time, these are the situations we deal with clinically day in and day out. So now I open the forum for questions. If you have any questions, can please leave in the chat box or please feel free to unmute and ask. Okay. So thank you
I
again.
can ask the audience, are any of you practicing at centers where you do surveillance for multi-drug resistant organisms in either your solid organ or PMTs? And are you using those results?
So anyone in the audience, Dr. Geetu ma'am?
Looks like not, but yeah, we don't do it at my center either. We certainly use a lot of the MRSA NAIRS, and that's been very helpful to de-escalate bank use. But we are also not currently doing any universal screening for multi-drug resistant organisms, but we do have a cohort of patients that come from nursing homes and have really high healthcare exposure. So in some of them, we do Candida auris screening or certain specific types of multi-drug resistant screening.
Okay. So if I have to tell from our center, even we don't screen, but I do know certain centers in India do screen for it, though they will not be treating as such at that point. In case the patient goes into febrile neutropenia in the future, so they'll be using those results for initiating the empiric treatment. So otherwise, like in our center as well, we don't screen. I'm sure if we start screening with a lot of kids, so it is like way of avoiding the
How are you de-escalating or reducing antibiotic durations in these patients with bacteremias? Because with our center, despite the data, it's been hard to convince our colleagues to be shorter courses. But I think some of this literature is definitely promising. So
yeah. So yes, I mean definitely no. It is very difficult, especially in India. It is very difficult for them to stop, even though patient has become febrile. I felt when I visited in some of the European countries, they are quite strict in their policy and they are following they 24 hours of febrile. They stop the antibiotics, but in India, not that possible. Even though they till complete recovery of the neutrophils and they still believe in the days, 7 days, 14 days. So yes, very difficult. So if there are no questions, please, I think we'll end the session here. Thank you once again, Dr. Neerja. I hope, and for joining at such a odd hours. So if please be, if you want, you can leave the session out so that you can take rest well, or I will. Yeah, thank you.
Hey, before you sign off, Dr. Nirja, you may probably want to share the link to the on-demand for those who are interested would be able to look into
Yeah,
that
certainly.
IDweek,
Yeah, yeah,
that would be
I'll put
great.
it in the chat now.
Yeah,
yeah.
yeah.
So for those who have attended anytime the ID week, so they keep sending you reminder mails regarding this access. And even if you have not registered prior, you can still register to take this access. So I would definitely recommend. So now we'll move on to the next talk, which is quite interesting. I really wanted to meet sir in the ID week, but then some clashes with the schedule, and I couldn't meet sir. So today we have with us Dr. Manish Paul. Sir is the is a distinguished clinical microbiologist and an innovator in the field of anti-infective drug development, and he's a co-inventor and the program lead of Enmetazobactam, the first anti-infective molecule invented in India to secure approvals in US FDA, EMA, Indian CDSCO in 2024. And as we all know, we are India is the hub of multi-drug resistance. So sir has brought us this new molecule, which is going to be, I hope, a game changer. And apart from the being this, he has conducted significant research in various other novel anti-infectives, and he is a postdoctoral, received postdoctoral training in infectious diseases at St. John Hopkins School of Medicine, and also a ORISE fellow. Uh, and he has been a part of advisory to multiple pharmaceuticals and medical device companies. So today we are here happy to hear the success story of the Enmetazobactam through from the inventor himself. So again, thank you for being a part of this small initiative. Sir, over to you, sir.
Thank you, Akshata. It's a pleasure to be part of this conversation, and yeah, of course, you wouldn't meet me because you would anyway meet me in India, of course, right? So we could hear this story once again here rather than missing out some of the sessions there, which are very interesting. And Neerja put it very well. The the kind of data she has collected and put across in a concise manner for everyone's benefit is really an amazing job she's done. But I would go straight into my topic. Let me put up my screen. Yeah. So I would put in a disclaimer here because here I'm speaking in my capacity as a scientist than representing Orchid Pharma directly here. This is the presentation I made at Bug Hub stage on 18th of October. Discovery of Cefepime, actually discovery of Enmetazobactam, but the drug combination is with Cefepime. I did work at Orchid Pharma, and I continue to lead the phase 4 clinical trials at this point in time. Well, you could see that here, invention to approval, the journey takes 18 years. Now, all of you, I think most of you who are participating in this conversation today are clinicians. And for you, treating a patient could be 7 days or 3 days or up to 21 days, or some very rarely would go in for long-term treatment of anti-infectives. But look at the journey it takes to come from ideation till to the clinics. It's a really arduous journey. But do you know who is the one who chases down these antibiotics and makes them inactive and useless? It's not the anyone other than the microbes themselves. Because in as much as we as human beings want to exist on this earth, so does microbes also want to live. So this molecule was invented in Orchid Pharma, and I came back to India from US FDA in 2006 and joined the group. And this program was conceived by the Dr. Om Reddy and others in the team. And Dr. Senthil was my colleague who was the medicinal chemist, and I was the lead biologist who led this invention phase. And for the discovery phase, on the developmental phase, of course, we had several other scientists whom I'll mention later. But when it came to phase 1 ready, we did out-license it to Allegra Therapeutics because it's very difficult for any Indian company to bring it until the clinics. It requires a lot of money, a lot of investment. But having a partner, in addition to financial benefits, also has a reach over to the global market so that we complete the global trials much faster. So Allegra in-licensed this molecule, and then they conducted all the 3 phases of clinical trials. And Indian territory is left with us at Orchid Pharma, and so that is what we are now leading it. So as I put it here, US FDA, that's a pride moment for us, right? So I put a slide here which says, this is journey, of course. I'll skip some of the slides. I put this Indian flag wherever I went. I was at Gordon conference also earlier this year where I presented this, the same slide, that it's a pride for all of us that we are able to finally deliver a molecule that US FDA has approved, right? Usually we in license or generically manufacture out of patent molecules and sell them. But this is the first time we have something where we have invented in India, right, as a group of scientists, and we have stood by it all these years and brought it to the market. So that's a pride moment. Of course, the second company which we have in pipeline is Workhard, and then we have Bugworks, who are also working at phase 1 level. But they would also, we hope they would come and reach the clinics also. We had several drug discovery programs, but the green one is what we call it improved chemical entities, where we looked at developing a beta-lactamase inhibitor. The rationale being a discussion which happened between the medicinal chemist and the biologist is Senthil was my partner. And what we thought is attacking a bacteria is very important, but at the same time, figuring out how to inactivate its defense mechanisms, because the ESBLs were evolving significantly. And I had just come back and I had a network of scientists. I was working there and I thought, let's expand this program in a different way. And we got some strains from abroad and we implemented the program in a very different manner altogether. And in the development of the molecule, we had several people who came in, like Dr. Sridhar Narayan, Dr. Sriram, who led the program. And we made an amazing culmination of events with business development team and everybody. So this is a picture which shows how many people get engaged at various stages. And it's like, it's several lives what a scientist lives in this, because identifying the disease comes from the discussion with the clinician. And then among the scientists, we come about discussing with the chemists and the molecular modeling people. Now it is AI. We come up with some targets and then we make these molecules. The medicinal chemists synthesize these molecules and give it to us as biologists. And what we do is when we evaluate its biological activity, we just say within 24 to 48 hours, this molecule is inactive. So the weeks and weeks or sometimes months of work what a medicinal chemist has put up just goes down the drain because it did not show activity. But it doesn't stop there. We go there and look at that molecule and make iterations. We make some more changes. I'll show you one slide about that. So as we make changes, we see any improvement in the target binding. Target is in the bacteria, right? And then once we have that, we come to identifying a lead molecule. Again, we start fine-tuning the program, and then we have clinical candidates. So in a BLI, we have 7 criteria. It should be specifically targeting the beta-lactamases. It should have broad spectrum of beta-lactamase inhibition, right? And then it should imitate the beta-lactam binding, because that is how the structure of the enzyme and the molecule will go and dock into it, right? And then we look at the PK, because drug-bug interactions and body-bug interaction, body-drug interactions all have to be borne in mind here. Penetration is an important aspect so that it reaches the site. And then it should not induce antibiotic resistance. Otherwise, what happens very soon, the antibiotic becomes useless. Now, these are the 2 patents which are the basis on which it is standing. One is the composition of matter, what we call it, the first patent, what we made. You can see the list of inventors here. And then we went on to improvise that molecule and then made the new set of method of use. So this molecule actually has anti-KPC activity, okay? The second patent which we published is in that regard. Now, when you look at this molecule, it just looks similar to Tazobactam and Tazobactam, right? But when we add a methylation here in this particular part, that's when it becomes Enmetazobactam. It was earlier known as OCID 5090, that is Orchid's infectious disease identification number as 5090. And AAI 101 was Allegra anti-infectious molecule, the in-licensed number. You can see in literature, these 2 numbers will be visible mostly before it got the name Enmetazobactam. And why it has relevance is when you look at cefepime along with Enmetazobactam, you will see even cefepime has this kind of a ring which is similar to Enmetazobactam. Now, this is called as zwitterionic ring. So when it's positioning, when there is methylated group of zwitterionic nature, both penetrate much better into the periplasmic space, into the cell. So that is very important for us to make PK parameters balanced. All right. But and at the same time, if you look at cefepime, it has got inherent AMC stability, right? Whereas enmetazobactam does not inhibit AMCs, but when you use it with cefepime, you don't need that function because it is inherently stable to it. So this is what I'm highlighting. Zwitterions are able to penetrate the gram-negative cell wall at a faster rate. And due to this nature, both can work better in the cells. So this is a graph which shows, I'm trying to make it very brief because not to bore you too long, because I've gone short time. And in this, if you see the inhibition curve, Enmetazobactam, if you look at the IC50, the inhibition concentration, what we need is very less compared to Tazobactam. Now, what does this signify? Now, you will see here among all the various enzymes what has been tested and published, you will see IC50 for Enmetazobactam is significantly lower than Tazobactam. This tells us, yes, it is active against ESBLs, KPCs, and oxa like beta-lactamycin in vitro. Why does that happen? These are the molecular structure, modeling structures of the enzymes, the colorful ones. And these line diagrams are the molecules of Enmetazobactam versus Tazobactam. So the number of sites where Enmetazobactam goes and locks into the enzyme is multiple compared to Tazobactam. That is the reason it is a very strong binding. Now look at these numbers on the left side. Enmetazobactam, the number of units required, that's we call it turnover number, is so less compared to Tazobactam. That means when it goes and locks in, you don't need too many molecules of Enmetazobactam to keep the enzyme inactivated till we allow cefepime to go and kill the bacteria. Right? Whereas Tazobactam, it keeps circulating very fast, in and out, in and out, in and out very fast. So you need more of the compound as well. And during that process, that in and out time, if it is more, probably, there is a chance that we allow microbes to start evolving further on. This is the one I was referring to, what iterations we make, right? Every structural iterations we go on making, it's too complex, but finally, if you see how the MICs keep changing, every iteration MICs went on changing. And finally, we have here Enmetazobactam, which has got the best of the activity. This is how we come to identifying a lead molecule. SAR is a structure-activity relationship, what we say. And again, from the published data, you will see against various number of strains, around 7,000 strains were tested. And when we look at the activity, cefepime Enmetazobactam, and if you look at ceftazidime avibactam and meropenem averobactam, you will see all of them are having, and meropenem alone also, if you see, it is very close. Now, this is an extended spectrum beta-lactamase specific molecule, right? It is, so does it behave equally with others? Yes, it does. So what we did is we looked at some of more data to see how the overall susceptibility comes. You will see it is up to 98%, so 97.7 to 98.8%. Pretty much it is very highly active combination which one can use. This slide is very interesting. Now you look at the column with meropenem activity. They're from two different studies, okay? You will see 96.2% to 97.6% is the susceptibility of ESBL plus enterobacterial strains, right? ESBL producing strains. Now, when you compare that with cefepime and enmetazobactam, it is comparatively better in this published data. So which tells, now we can use this as a carbapenem or a meropenem sparing molecule in the clinics. We don't have to reach out to a carbapenem as the first line in critically ill patients in the ICUs with complicated UTI or HAP or RAP. We can first use cefepime and enmetazobactam. And the approval is for this molecule, this composition itself right now. All right. Then the turning point in the whole journey, what made it a different thing? What makes this molecule as a unique molecule? This is mainly due to the in vivo model what we developed. The in vivo model was the one which changed the game of entire life of Enmetazobactam. Until then, it was going in a direction where we were struggling. And when we found a novel model wherein I think it's too much of discovery space. I'll just tell you simplistically, we try to increase the concentration of enzyme in the body of the mice, and try to give a slow release by making a soft gel agar into the and keeping it under the flank of the skin and under the neck and on the neck of the mice, so that it acts like an infusion, and we'll see how the it pans out. And we see that, look at these numbers. This is a combination with various antibiotics, and you will see the activity is much better compared to that with Tazobactam. This is how we show the differentiation, which was telling us, okay, this composition is definitely better than what is existing in the market. It is not that Tazobactam is useless. Tazobactam is becoming resistant to many of the bacterial strains producing ESBLs. That's why we are going for meropenem. So we could now look at a newer alternative. Now, how do Orblicef is the trade name of Orchid Pharma. Orblicef simplistically means orchid beta-lactamase inhibitor with cefepime or cefepime. Right? It is useful as an carbapenem sparing molecule. Now, everyone would have this question. We have Zidibactam also coming in, cefepime Zidibactam, which is a better one? I think both have to be looked at in a different way, because side by side, the comparison cannot be done, because Zidibactam and Tanirobactam is the other one, were designed to overcome resistance against class A, B, and D. And Zidibactam also has an inherent antibacterial activity, while Enmetazobactam is a pure BLI. So it is not like two antibiotics and a BLI going in there. It is just an BLI coming and restoring the earlier antibiotics. That's the function of Enmetazobactam. Now, these two slides are to show you how we made the decision to take it as a drug. Because it was working on ESBLs and KPC, we said, yes, there is a emerging infections. This is going to work. Then we looked at various types of enzymes it was working on, and it was not inducing AMC. That is another thing. If it induces AMC production, then we have another challenge. Even that was taken over. And then we also saw that it has got excellent PK parameters. I'm almost coming to the end of the presentation on 3 more slides. I will show you the phase 1 PK study. The preclinical PK was so beautiful. We wanted to see whether it shows the same thing in vivo in humans. At this time is when we out-licensed it to Alector Therapeutics. And the penultimate bullet is important. We had to test against various off-target interaction of the molecule in humans. It's called PanLab panel, because multiple human targets are there, and it is hit against that various targets to see that it doesn't have cross-reactivity. That's how we evaluate safety and side effects. Then once it is out-licensing is done, the clinical trial outcome is the red letter is the one why this was approved by EMA without conducting a clinical trial against HAP and VAP. Because if you look at the AUC of the plasma of cefepime and enmetazobactam is almost similar in healthy volunteers. When they saw that the lung penetration is so good, so why don't we approve it? So without this any trial, it has been approved for HAP and VAP. Then in phase 2 studies, we look at the efficacy, and we showed that 500 mg was the one which was used of enmetazobactam with 2 grams of cefepime, and it worked pretty well against cefepime-resistant Enterobacteriaceae. So these strains which the patients had cefepime resistance, and against them this was used. And phase 3, of course, we had to do a larger study in multiple centers, and total 112 study sites were there in 19 countries. And the study showed amazing superiority. Superiority because earlier it was positioned as a non-inferiority trial. But then if you look at this graph, the eradication and clinical cure rate was much higher, because of which this has been given as a converted into a superiority trial, and it got approved. Final snapshot, Orblicef. It's a fixed dose combination of 2 grams cefepime with 0.5 grams enmetazobactam. Unlike there is one composition available now, cefepime tazobactam, which is 2 grams each. So the concentration of drug is also less. Complicated UTI, including pyelonephritis, and additional indications in India, EMA, and UK MHRA, but not in US FDA, is HAP, VAP, and bacteremia due to these conditions. Activities against, as I told you, against all the strains, E. coli, Klebsiella, Pseudomonas acinetobacter, against STEM, SHV, CTXM, and MC producers. It's a 120 minutes IV infusion every 8 hours for 7 days treatment, and superior efficacy compared to peptides, right? And safety tolerability is similar to the other beta-lactam classes. So whatever hypertension or a hypersensitivity in reactions, what we look for any beta-lactam class of molecules are the ones we have to bear in mind. Though the positioning is a first line therapy, we have to await the regulatory approval for that. But it is definitely based on the data. It is a carbapenem sparing molecule. That's where I end my presentation, and I would like to take any questions if you have.
Thank you, sir. That was really a remarkable journey. So it gives us hope, and for like a molecule discovered in India for specifically for Indian infections, I would like to say. Uh, this work exemplifies how the innovative science and determination and global collaboration will lead to development of such life-saving therapeutics. Definitely, we have a dearth of antibiotics. So, I mean, recent past, like we have having some hope against these MDROs. I think there was one question, sir. Like I think cefepime, whether it will the Enmetazobactam efficacy against AMC. I think it was obvious. So would you like to take the question?
The question is whether it has got activity against cefepime.
Against AMC.
Uh,
AMC
okay.
produces it.
Okay. Well, yes, it does take because cefepime itself is active against Enmetazobacter against AMC producers, right? So the question doesn't arise anymore that whether it is one moment whether it is active or not, because Enmetazobactam is not an inhibitor of AMC, but in combination with cefepime, which by inherently is active against cefepime, combination of cefepime, which is inherently active against AMC, we don't have to worry about AMC producers. And at the same time, Enmetazobactam does not induce the production of AMCs. That's another aspect we have to bear in mind. So combination-wise, it is amazing against AMC producers.
Yes, sir. Thank you, sir. Sir, also I would like to ask one question. So
Sure.
most of these, I think I'm sure it will have activity against the MDR, like TREs and MDR, I mean, metallobetalactamases. So whether those studies are planned or I've seen most of these trials, everything stops at the level of comparing with miropenem or the BLIs, older BLIs. So is there any study planned or is it already there?
Okay, I'll tell you. Now, if you see, this is an extended spectrum beta lactamase inhibitor, ESBLs, specific molecule, right? It doesn't work on metallo-beta-lactamases because in metallo-beta-lactamases, it is zinc. It is the combination is the way the enzyme is made is very different compared to ESBLs. ESBLs are serine beta-lactamases. So the way the molecule goes and locks in is very differently here compared to the metallo-beta-lactamases. So in other words, it does not work on metallo-beta-lactamases. That was very specifically when you suspect MBL infection, you got to look for MBLIs there rather than come for this one. But in most of the hospital infections, if you see the rate of infection also, it is usually ESBL producers, that too which are now resistant to piperacillin and tazobactam. That's when, as an ID person, you reach out to a meropenem first line. To when you know it is not working on peptides, you directly go to Mero. You don't have to go to Mero. You can directly go for Enmetazobactam with cefepime so that we can still save the carbapenem for when it fails. Even at this stage, you can go for carbapenems.
Okay. Okay.
Right?
Yes, sir. Thank you, sir. So, importance of zwitterion in penetrating in the GNB. There's one more question, sir, if you would like to take up.
Yeah, I mean, the way the zwitterions bind with this, the cell, all these molecules and everything, the way they work is they attach to the cell wall and go penetrate and go into the cell, right? It is all, everything happens through ionic combinations and recombinations and breaking off. That's how everything moves into the body, right? Every cell wall. That's a different lecture I can conduct sometime where I can show you how each mechanism of action of all the 5 types of mechanisms of action I can explain to the audience. Drug resistance, how bacteria produces and how each antibiotic functions. I can give it a separate lecture for you guys if you want to some other time.
Okay. Thank you, sir. Thank you so much. Like we discovered the challenges in the drug discovery from preclinical research to reaching the stage of clinical trials. Now we understand why we are not having much of the newer antibiotics in the pipeline. So
That's
all 18 years of efforts, finally, at least it has bore fruit and really we are proud. Like we have made us proud as an Indian for bringing out such molecule which has even got a approval from the US FDA. Thank
Thank
you
you.
once again, sir.
You're most welcome.
Uh, so with that, we'll close this session. So like let's start the next and the last session.
Okay, I just left my email address. If anyone is interested to discuss more, you're most welcome to reach out to me. All right.
Yes. Thank you,
Thank you.
sir.
Thank you.
Is my is the screenshot? Is my screen visible for everyone? Okay. Is my screen visible?
Yeah, it is.
Yes, 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 it is.
Okay. I don't press any mistake. Okay. Yeah, so that brings us to the last part. So again, the disclaimer, most of these slides which I have taken is from the IDweek presenters there. Like there were two, the IDweek is practically very huge with more over 250 sessions, like which 250 sessions which had the presentations, and there were other stages like Global Hub, Bug Hub stages, which again was a mine of like various knowledge sharing forums. So it is humanly not possible to recapitulate all of this. So I've tried to present the studies which I have felt was interesting or which could be practice changing. I've tried to organize it, but again, let's dive into it. First, I'll be talking about the antimicrobial stewardship. So I think balance trial was one of the trial which was like first spoken about in the IDweek and could be practice changing. So this balance trial evaluated 7 days versus 14 days of antibiotic treatment for patients with bloodstream infections. So it was an RCT which was conducted in 7 countries, and it had around 3,600 patients. So they compared 7-day course of appropriate antibiotic versus 14-day course of appropriate, and looked at primary outcome was all cause mortality, and then secondary outcomes they evaluated was recurrence of bacteremia, relapse, adverse effects, length of hospital stay. So the results which were quite promising was that 90-day mortality amongst the patients with 7-day and 14-day where both of them it was non-inferior, and the risk difference of minus 1.6%, and it was 14.5% in 7-day arm versus 16.1%, which was not statistically significant. There was slightly lesser length of stay in the 7-day group. But important things to take is the exclusions, which were important exclusions where the staph infections were excluded, fungal bloodstream infections were excluded, and also patients with severe immunocompromise and prosthetic heart valves were also excluded. But still, I think this gives us hope that shorter is better. Next, we have the INSPIRE trial. It was a 92 hospital cluster randomized trial of intelligent stewardship prompts to improve real-time empirical antibiotic selection versus routine antibiotic selection practices for patients with skin and soft tissue infection. Basically, what they did is uh, they compared the standard of care of routine antibiotic stewardship, which all of us do, with the intervention where there was a computerized physician order entry. So by it was a bundle. So there were certain prompts which were there asking for the various parameters of the patients, and based on these prompts, the antibiotic was given. A choice of antibiotic was given, and it was real time, and patient specific risk estimates were taken, and based on this, the guidance for usage of these extended spectrum antibiotics were chosen. So outcomes were quite good. So this led to 28% reduction in the use of extended spectrum anti-pseudomonal and anti-pseudomonal antibiotics. and there was no safety or safety breakouts, or the length of stay was not significantly different between the two groups. So this could be an practice changing update, because I'm sure most of your hospitals will not have ID experts, or even if they are, there may be few in number who cannot take care of the entire hospital, and or most of the like it will be based on consultation basis. So this hospital based stewardship practice of in starting this computerized order entry would prompt reduction in the usage of wrong antibiotics. Next, we have an update in the diagnostic stewardship, that is UL core BSI test. It's a molecular testing for pathogen in the blood directly from the blood. So we have various directly from the blood tests, which includes obviously your biofire, and then there is also T2 T2 MRI. So this is one new platform. So it is rapid within one. The TAT is supposed to be less than 1 hour. This it is easy directly from the blood sample, and it will detect the total microbial DNA, and it is broad based, and they have even the 12 resistance determinants, and very less amount of blood is required. So what is the major difference in between this as well as the biofire, etc.? So here they take into the total microbial DNA, including the viables, non-viables, and microbial DNA. So, and what is novel is the inclusion of the different and organisms. So if you can see, we have all the clinically significant gram positive, clinically significant gram negative, including the anaerobes, and detection of antimicrobial resistance, which is of importance, including your MBS testing, and for staff, the VAN-A, B, and VAN-A, VAN-B, and MEC A and C testing. So we require very less time and very low volume, and it had given good results in the preliminary test. Only thing is, I'm sure the cost factor, and further they are evaluating for the sensitivity for detecting the various organisms as well. So next, I'll be talking about hepatitis C. Hepatitis C, the directly acting antivirus for the DA or the DAS for the hepatitis C, has been a revolution in the management of HCV. But one caveat here is, like there is a specific guidelines or FDA approval for treatment who are in, except for those patients who are in pregnancy and early infancy. There is no treatment which is approved for early infancy up to uh, 2 years. So that is why there were trials like HIP-1 and HIP-2, where they looked at the pharmacokinetic studies of the DAs, that is ledipasvir, sofosbuvir, and sofosbuvir velpatasvir in pregnant women. So basically, it is said that they are not, no guideline is advising DA treatment in the pregnancy, because one thing, there can be spontaneous clearance of this HCV. Second thing is because of the deranged pharmacokinetics, so which could reduce the levels. So this was a study design where they enrolled the pregnant women in the gestation 23 to 25 weeks, and they have taken down started the treatment and taken the blood samples for the PK as well as the viral loads, and looked at the adverse effects, and also looked at the sustained viral response after 12 weeks visit. So the HIP-1, there was recruitment of 29 participants, and HIP-2, there was recruitment of 14 participants. And one important thing is, those who ever completed the study, they had attained SVR, that is the sustained virological response. And this is the pharmacokinetic data. So they have looked at the sofosbuvir, ledipasvir, and velpatasvir levels. So, and the GS331007 is the nothing but the inactive renally excreted sofosbuvir metabolite. If you can see, there was a reduction in this inactive metabolite only, but others were pretty the same. And when they looked at the HCV viral response on the treatment, all the mothers, the 18 mothers who were treated, who have completed the treatment, and we had they had evaluated for 12 weeks, the SVR was achieved. So conclusion is that there is no clinically significant PK changes. So we can, though the inactive sofosbuvir metabolite was reduced, it is likely due to the increased GFR and unlikely to be clinically significant. And so this also reassures the safety and efficacy of these drugs during the pregnancy, though larger study is advised. Another study which was presented was the STORK, that is safety, tolerability, and outcomes of velpatasvir sofosbuvir in treatment. So again, they evaluated the SVR after completion in the during the pregnancy. So they enrolled at 20 to 30 weeks of gestation, and they have looked at the HCV viral load as well as the adverse events, including the neonatal adverse events. So the outcomes were promising. So the total number of participants, though, were 24. There was only preterm births, which were in the three only preterm birth, which was mostly due to the other issues rather than the HCV induced or the treatment induced. And all the patients achieved the SVR at 12 weeks. So another study, this is another study which is planned, is the looking at the pharmacokinetics and safety of glecaprevir and pibrentasvir in this group, that is the impact to 2041 study, which is just started and is expected to complete soon. So why is all these studies important? That one thing is, so we don't have to worry about the PK parameters, so we can use the sofosbuvir based regimens which does not require any adjustment in the pregnancy, and the safety was evaluated and it was found to be safe. Other thing, data gap is most of these studies which looked at the treatment for HCV in pregnancy was beyond. Most of the studies was beyond 20 weeks. So less than 20 weeks, that is around 16 weeks, we don't have any study evaluating. That is so that is during the organogenesis period. So we don't have the safety data for during that period. And and also they are also commented on various other off-label treatments. That is, they have the registry of all the patients who were treated off-label, which also showed good response as well as no safety danger signs. So definitely this would help in treating the patients during the pregnancy and avoid the further transmission and the complications. And another thing which we don't know is the PK PD of the directly acting antivirals in the breastfeeding. So next, I'll be talking about the new outbreaks. So one which was discussed, which was selected, was the Chandipura vesiculovirus outbreak, which was in the outbreak in India, which happened in the Gujarat. But one thing, why I chose this was the highlight was there was though basically Chandipura vesiculovirus presents as encephalitis syndromes, but when they during the study they found that this outbreak was different because many of these patients, apart from having the usual encephalitis like features like fever, altered sensorium, there was more of MODS like picture, including patients presenting as failure, sepsis, septic shock, and deranged LFT and coagulation profile. And the authors also made a comment that such high numbers of systemic involvement were not known prior to this outbreak. And especially in a situation like India, which is a tropical hub, so where we have a lot of tropical fevers, which can, like including dengue, which can mimic or which can present to us with fever and MODS-like features or thrombocytopenias and deranged LFT. It is likely that the Chandipura virus will go unnoticed, and hence the diagnosis time to the diagnosis also will be higher. So uh, uh, most of these patients presented as hemorrhagic fever overlapping with encephalitic features. Hence, we should have high suspicion of Chandipura virus, especially in the outbreak states like Gujarat, especially in the high peaks like June to August. Next, obviously, the outbreak of interest is the mpox. Uh, as we all know, the only license or the approved treatment for the mpox is tecovirimat. But again, this was approved for mpox because this was approved for smallpox. This is the study results of the PALM 007 trial, that is outcome of it was a RCT, randomized placebo controlled trial in the Democratic Republic of Congo, evaluating the treatment of tecovirimat for mpox. They looked at the clinical efficacy, which was assessed as the time to lesion resolution in tecovirimat plus standard of care versus placebo plus standard of care in patients with mpox. So it was a 1:1 randomization, and the patients were usually hospitalized for the 14-day treatment duration. Another important thing is tecovirimat. The drug levels depends on the status of the nutrition. You have to give it with a nutritionally rich, high-calorie meal, including fat. And as we all know, in Africa, there are a lot of nutritional deficiencies. So they gave it with along with adequate calorie intake for the drug absorption. And they took a blood and OP and the lesion swabs every other day. And this was the assessed as well as the PK the parameters. Next, the primary outcomes, there was no statistically difference in the time to lesion resolution between the intervention and the placebo group, and including the mortality rate, there was no significant differences. No differences were observed with any sample type by the treatment, whether the patient was on placebo or the tecovirimat. So the conclusions were there was no, though there were no safety concerns, but there was no overall reduction in the duration of treatment even though patients were treated with tecovirimat, and there was no difference between the virological outcome. Only thing they noticed was when they did the subgroup analysis. So they had in the Democratic Republic of Congo two subgroups. One was the hospital in Kole and one more in Tunda. So there was some differences where there was more treatment effect seen in Kole but not in Tunda. So which they have yet to analyze it further, like which subgroup will benefit from tecovirimat. Next, other ongoing trials for monkeypox, we have a lot of trials: STOMP, Unity, Canada, Epoxy, and these are all trials with your tecovirimat. Other drugs which can be used in case of mpox, one of it is brincidofovir, which has been used again it was approved for smallpox, and that is why it has been approved even for mpox. There's a MOSA trial which is planned for clade 1 to evaluate this brincidofovir. Next, just a brief on the vaccination. So basically, the vaccination, the vaccine Genos vaccine, is usually recommended. It was clade 2 specific vaccine, but there is some sort of cross protection which is expected, and there were few FAQs which was discussed. But I think because we don't have much of this vaccine and the treatment is available, I'll be just skipping that. Next, staphylococcus infections. I'm sure this is like the favorite topic for any ID. So one thing which was discussed is the long-acting glycopeptides, the newer ones, the dalbavancin. The DOTS trial, it was also already published prior, but it was also discussed in the IDweek. So basically, that is dalbavancin as an oral option for treatment of Staphylococcus aureus bacteremia. So here they compare 2 weekly injection of 1,500 mg infusion for more than 6 weeks, and why it is supposed to, and it has a long acting because it's very highly protein bound. So this trial was an open label trial where they randomized patients to get dalbavancin or standard of care. The standard of care, depending on MSSA or MRSA, it could be cefazolin, nafcillin, or for MRSA, vancomycin and daptomycin. So it was after the induction, in the initial induction antimicrobial for up to like at least 4 days or up to 10 days, followed by giving the dalbavancin or the standard of care. So the key exclusions were uncomplicated bacteremia. So basically, they targeted the complicated SAB. So they excluded uncomplicated bacteremia, CNS involvement, left endocarditis, and retained prosthetic materials and severe immunocompromised patient. Another important thing about this trial is the usage of this door, that is desirability of outcome ranking. So this is also one of the way of interpreting the primary outcome. So desirability of outcome ranking, that is has ranked like 1, 2, 3, 4, 5. So in that, based on whether the parameters like patient is alive, or how many of them had clinical failure, infectious complication, or the adverse effect relating to the drug discontinuation and the quality of life. They have analyzed the results. So here, if you can see, there was no difference between the standard of care or the dalbavancin in terms of the mortality or the adverse effects, then as well as the clinical failure or any complications. So it had a similar risk or benefit in compared to standard of care, but it was not superior by door. It was non-inferior by the clinical efficacy, and hence we can use, it's a viable option for complicated SAB. So what are the recommendation? You can use single dose of dalbavancin for uncomplicated Staph aureus bacteremia when they have less than 14 days, or in case of complicated Staph aureus bacteremia with adequate source control, and skin and soft tissue infection where other options, oral options like Clinda, you cannot give Clinda, or linezolid, you can go for that. Then 2 doses is recommended. Where do you need to give 2 doses? Is when that is first dose at day 1, second dose at day 8. You can give when in complicated Staph aureus bacteremia, vertebral osteomyelitis, sternal wound, and better avoid this in case of CNS infections or epidural abscesses, and infections in which source control cannot be achieved. So any undrained abscess, any processes which you cannot remove, and obviously in the polymicrobial infections. Next, also there is oritavancin. So again, all these are similar in action, but again the mechanism of action, supposedly the dalbavancin has basically inhibits the transpeptation, whereas oritavancin is supposed to inhibit both the transglycolation and the transpeptidation, like your vancomycin. And the major difference, obviously, is the protein binding. The dosing of oritavancin is 1200 mg on day 1 and 800 mg on day 8. But the major difference is in the susceptibility of Enterococcus faecium, especially. So if you see these, the headings are vancomycin susceptible, Enterococcus faecalis, vancomycin resistant. So this one in Enterococcus faecalis, vancomycin susceptible, both of them can be used interchangeably. They have the similar MICs. In vancomycin resistant Enterococcus faecalis, dalbavancin had a very high MIC, so oritavancin would be preferred. Then in vancomycin susceptible faecium, again similar. Vancomycin resistant Enterococcus faecium, oritavancin again is better for Van A type of resistance. So again, oritavancin, multiple studies showed comparable efficacy. Most of the, but the limitation is most of the studies were retrospective. So again, when can these the issue again would boil down to the cost. So in India currently there is dalbavancin which can be used, but again the cost is quite prohibitory. So around it will cost around 20 to 25,000 per dose. So most of the patients would definitely prefer to be on like injectable drugs which can be or given cheaper, or the lower oral alternatives which are available currently. Next study is the EVRIOS, that is comparative evaluation of the low versus high doses of rifampin for treatment of Staphylococcus bone and joint infection. So again, we know in bone and joint infection, we do give rifampicin along with the any like depending on MSSA, MRSA, we will give either the cefazolin or the vancomycin. And also we give it once the culture like blood culture is sterile, and we give it because of the anti biofilm activity. So, but the dosing is always a matter of discussion. So the low dose was 10 mg per kg per day, whereas high dose was 20 mg per kg per day. So they looked at the failure defined as persistence or recurrence of infection after within 1 year of the completion of the antibiotic. Secondary outcomes were the adverse effects. So the results showed there was the low dose versus the high dose. There was no clinically significant difference when I analyzed by ITT as well as per protocol. And the serious adverse effect was little more in the high dose group rather than the low dose group. So we can safely use low dose rifampicin 10 mg per kg QD in the bone infection, and it has a high success level as well as less side effects. But only thing is which it does not answering is whether we can use shorter regimens or when to introduce. So where should we introduce early or wait for the inoculum to be low for giving the low dose of infection. Next important ones which I want to discuss about is the lung infections. So empyema, again, there are a lot of cases of empyema. So it is always a matter of contention for the duration of antibiotics in empyema. So there are various guidelines which ATS, that is American Thoracic Society, is recommending 2 to 6 weeks, of at least 2 weeks from drainage and being afebrile. So, whereas British Thoracic Society, which is 2010 guideline, will recommend prolonged course and at least they recommend at least 3 weeks of therapy. And ERS ETS statement on pleural infection, they have no specific recommendation, but they do mention that at least 3 weeks. So one of the scores which can be, which was identified was a rapid score. So it is based on the post hoc analysis of 2 RCTs, and they have put them in the patients in the 3 bucket: low, medium, and high risk. So the rapid score includes your blood urea nitrogen, age, purulence of the pleural fluid, infection source, and the serum albumin. So based on the score, you give a 0 to 2, 3 to 4, and 5 to 7. So you'll be putting them in the low risk, medium risk, and high risk category. And based on this, so higher the risk, you'll have to give a longer duration. Low risk patient, you can still go do away with shorter duration, that is up to 2 to 3 weeks of therapy. Then bronchiectasis, again, this is like I am sure most of the patients with this bronchiectasis, it's always a dilemma whether to give the antibiotic, not give the antibiotic. So here, this is the recommendations which they have put forward. The first line therapy will always be airway clearance. So there are various modalities. First and foremost should always be giving the respiratory physiotherapy and the airway clearance. Anti-inflammatory, that is the inhalational corticosteroids, we should always use with caution. Some of them, many guidelines also do mention about the maintenance antibiotics for frequent exacerbator, that is use of azithromycin mainly for the anti-inflammatory action. So we again we should come with multiple cautions, like you should rule out the patient should not have any NTM infection, and after trying all the first line therapy, then only you can give the maintenance antibiotics. Then they have also discussed about the inhaled antibiotics, especially for patients who are suffering from chronic pseudomonas infection. So in that, the best data is available for aminoglycosides and the colistin. And one more important thing is when do you, during the period of exacerbation, whether to like how long do you treat, and eradication of the targeted organism, should it be or not. So these were the things which they had discussed. On what routinely should be avoided is the routine use of all corticosteroids or oral steroids, routine use of antibiotics. So next important thing is inhaled antibiotics. So when do you give the inhaled antibiotics? When there are 3 or more exacerbations per year, and this evidence is mainly for Pseudomonas infection. So again, what should be considered is whether the patient should be doing the first line management, that is the airway clearance, after which certain chronic infection should be ruled out and it should be individualized. So the data is available majorly for aminoglycosides and the colistin. One of the study was the recent the study which was discussed was inhaled colistimethate sodium in patients with bronchiectasis and Pseudomonas aeruginosa infection. That is the PROMISE 1 and 2 trials. So which they showed that the inhalational inhalation colistin was quite effective in preventing the annual exacerbation of the CMS. Both of these studies showed promising results that this could be definitely an option for patients who are having frequent exacerbation. Next, moving on to the next topic, important one, HIV and TB. We had lot of HIV related updates, and they also discussed many of the updates from even the CROI. This particular study, the results of week 48 of the phase 2 study evaluating once weekly oral islatravir plus lenacapavir. So for those who are aware, not aware, islatravir is a nucleoside reverse transcriptase translocation inhibitor. So there was previously the results of week 24 was published, which showed that there was dose-specific decrease in the CD4 cells and the lymphocyte count. Though the PK modelings have suggested that it is not expected when 2 mg dose is used for the study. And lenacapavir, as we all know, is the capsid inhibitor. So giving this, it is we are going to look at the once weekly dosing, which will reduce the adherence issues. So this was a phase 2 trial which they compared the once weekly regimen versus the ongoing like standard of care regimen, which and the virological outcomes at the week 48 was non-inferior. It was almost equal to the standard of care. The CD4 counts, there was no which we saw in the 24 week. They had seen that there was a reduction in the CD4 count, but once they used the 2 mg dosing, there was no significant difference between the mean change in the CD4 counts at week 48. And so this islatravir lenacapavir has the potential to become the first weekly oral complete regimen for HIV-1 therapy. Then next thing is the long-acting cabotegravir rilpivirine plus lenacapavir in patients with HIV adherence challenges and INSTI resistance mutation. So basically, the newest US IAS USA guideline says that we can use long-acting cabotegravir plus rilpivirine can be considered in select patients with viremia, where there is like resistance to various NNRTI or INSTI. So this study looked at comparing the usage, the combination therapy. So again, this was it was found to be non-inferior. So other thing is there was no reported treatment emergent resistance, and but the thing is it was very small study population was quite low. So we have newer drugs in pipeline. This was again discussed in the AIDS 2024 poster. So we have GS 1720 and GS 4182. GS 1720 is a novel oral weekly INSTI. So we have various INSTI. This is the that which is the INSTIs which are given daily. So this is a novel INSTI which is once per week, and GS4182 is a novel oral weekly pro-drug of the lenacapavir. Again, this was a phase 1 trial which showed that they say it was safe and it was well tolerated. Another study which is ongoing is a wonder study. That is a combination of this GS1720 plus GS4182 in virologically suppressed. So this is a novel first in class where they are looking at the combination of oral INSTI plus capsid inhibitor weekly once therapy. This is also again this is a phase 2 trial, and it is ongoing. The next newer drug which we have is the MK8527. It is also a novel oral nucleoside reverse transcriptase translocase inhibitor like your islatravir. This is a phase 1 trial which is ongoing. Then we have the VH184, which is a third generation INSTI, which has broad spectrum of activity against all. And the in vitro potency remains same as the dolutegravir and cabotegravir. So we may be using it for long-acting regimens. Then artistry phase 2-3 trial, that is bictegravir plus lenacapavir in patient with HIV switching from complex ART regimen. So this is one of the trials which again is using lenacapavir for those patients who are on complex regimen. So the trial to the efficacy and safety results were showed it was non-inferior to the standard of care regimen. So, and the next novel regimen or novel treatment in HIV, which is the broad neutralizing antibodies. So that is using lenacapavir plus the BNABs, that is teropavimab and zinlirvimab. So basically, these are neutralizing antibodies, which can, when you give it in combination, you can give it once 6 monthly. So this one of the phase 1 trial was published. So this is the phase 2 which is ongoing, and the week 26 there was equal virological outcomes. There was no serious adverse effects. There was good viral suppression, and this combination, you can sustain the viral suppression for 6 months. So we reduced we are increasing the treatment duration, like reducing the number of injections which is required. So this can be once 6 monthly regimen if we it further approved. So in the prevention modality, what is new is there is oral MK8527 once monthly injection, which is currently in phase 2 2A trial, which is estimated to be completed by December. NTM, NTM is always on. So India, they had more discussion on NTM rather than the TB, because they have more of NTMs rather than definitely TB. First and foremost, the MAC. One important thing is the drug testing for the MAC, macrolide and amikacin is only recommended, because rifampicin and ethambutol, the MICs have not shown to be correlating with the clinical outcome. So the treatment recommendation for MAC lung disease, for nodular and cavitatory, we already know. So it is now 3 drugs for nodular, that which includes azithromycin, rifampicin and ethambutol. For cavitatory, more than so you can include even amikacin IV. And for regimen for nodular disease is 3 times weekly, whereas for cavitatory we need to give daily. One important thing which they've added is for the refractory in MAC infection, you give azithro, rifampicin, ethambutol with amikacin liposome inhalation suspension. So this was based on this study, ARISE study. So where they looked at adults with non-cavitatory lung disease or new or recurrent MAC lung disease who have not received antibiotic therapy for the current infection, and they randomized it into 2 arms. First arm is the ALICE arm, that is which received the liposome, amikacin liposome inhalation suspension, and along with the azithromycin and ethambutol, whereas the other comparator arm received the ELC is nothing but the placebo. So, empty liposome control along with the azithromycin and ethambutol was given, and there was so the patients with Alice arm performed better with early culture conversion at 6 months as well as the 7 months. Next, for the M abscesses, Mycobacterium abscesses. Again, as we all know, abscesses, we have abscesses, molleti, Boleti and mesylens. So one important thing is the macrolide resistance in M abscesses. So it can be of two types, inducible as well as your mutational. Inducible resistance is due to the ERM, that is erythromycin methylase gene. So presence in abscesses species, it's around 80%. Mesylens, it is less, it is almost zero. Boleti, again, most of these patients will have your inducible resistance to erythromycin. So another important thing which I got is like, first thing is obviously the clarithromycin susceptibility. You will look at the day 3 and 5 as well as day 14 to tell whether the patient is having the inducible ERM gene. So if it is susceptible, though, and the subspecies is mesylens, you will give macrolide gas susceptible and you can use macrolides for the treatment. But if it is any point resistant, it could be inducible or constitutive macrolide resistant, but you will include your clarithromycin or azithromycin in your regimen, but it is only for anti-inflammatory purpose. So you, even though it is coming as resistant, they are saying you include your clarithromycin or azithromycin in the treatment as an anti-inflammatory agent. So M abscess is based on the macrolide resistance. If there is macrolide resistance, initially you should give up to, you can even give more, 4 or more drugs based on, you can include two to three parenteral or 2 to 3 oral drugs, in which they say uh, you can include azithromycin for immunomodulatory action. So we have lot of drugs in pipeline for NTM infection. We are in phase 2, in phase 1 we have apramycin, phase 2 we have bedaquiline, clofazimine. So basically that is inhalational clofazimine. Then epetraborole, which was discussed in last IDweek. And then we have inhaled nitric oxide, inhaled GM-CSF, omadacycline. And in phase 3 is the amikacin liposome inhalation suspension, inhaled clofazimine. And we have also comparison of azithro versus clarithrol and clarithrol versus moxifloxacin, and also 2 versus 3 drugs per max. So these trials are very important for M abscesses, so which we can look forward for. And one interesting study which I wanted to highlight was Hanuman study. So yes, this is the Hanuman. So this was a Thailand-based study. So where they evaluated the immunogenicity and safety of high dose versus double dose quadrivalent inactivated influenza vaccine in adult kidney transplant recipient. So yes, it was, it was supposed to be included in Transplant ID, but I found it very interesting. So I just wanted to put it here, where they looked at and the vaccine strategy. So whether you use high dose or double dose. So high dose contains 4x the amount of hemagglutinin compared to the standard dose. So in the standard dose, it is 15 microgram, whereas in the high dose, we have 60 microgram. And this quadrivalent high dose formulation is available in Thailand, 0.7 ml. So the various strategy for against influenza vaccine is high dose, boosted dose, double dose, and standard dose. High dose is nothing but very high concentration of HA, as I mentioned. Boosted doses, you give standard dose at 1 month, and then again after 1 month you repeat the dose. That is your boosted dose. Double doses, instead of you give the standard dose 2 doses, that is double dose, and the standard dose is 15. So there are various studies which have compared between boosted, double, and double and standard. So there is no study which is comparing between the high dose and the double dose, which they are planning, they are doing here. So in the kidney transplant, they have randomized in 1:1 with to receive high dose as well as the double dose vaccination. So here they have seen that for at least one the seroconversion for at least one strain was greater with patients who are receiving high dose compared to that of the double dose. So they are recommending better to give high dose rather than that of the double dose. But again, it should be considered that with a pinch of salt because it was non-blinded and with the practical use of 2 doses of standard dose in real practice, and they have not looked at long-term immunogenicity and vaccine effectiveness in the real life scenario. So again, there was a lot more which I would like to discuss and I wanted to discuss, but again, I didn't want to bore all of you uh, in that. So I think that I'll stop it here, and if there are any queries, I'll be able to take it now. So I hope I have done justice to what I wanted to share here. Any comments or any questions? Okay. So I think that brings us to yeah, Dr. Neja, you want to add anything?
Oh no, I just think it was an impressive job done, whirlwind update, and it's a really tough thing to condense everything into
Yeah,
a short
there
time.
were
Yeah.
like really wonderful talks, which I'm like, okay, at least this gave me an opportunity to revisit. So I would like, I mean, in that way, it was a good exercise for me. So otherwise, I thought like it's humanly impossible to attend all the sessions, even if you are there at present. Like a lot of good sessions happening in so many different halls. Yeah.
Yeah.
So we have I again I think it would be better if you can all of you can register and access the IDweek virtual access, and it will require I think over 1 year you can sure like look at 2-3 sessions each. I think 1 year you'll definitely try to cover it. Yes. Thank you once again, Dr. Nirja, for being a part of this. So we disturb your early morning schedule on a weekend.
No, no. Happy to be here.
Yeah, thank you. Thank you once again. So I think we can end it here. Thank you all. Thank you for joining. So if for those who are on the Zoom, so all of this is available on my YouTube channel if you want to access it anytime. So there is one question. So for ESBL plus AMC producer, can cefepime Enmetazobactam be used? Yes, you can use, I think, because it has activity against ESBL as well as cefepime has intrinsic activity against your AMC. So if there are any questions, you can leave it on my YouTube channel so we can address it later. So thank you once again. Bye. See you all. Bye, Dr.